Forget looking at one gene at a time, painstakingly slow.
Yeah.
What if you could suddenly see basically your entire genome's activity all in one go?
Yeah.
That kind of radical shift, seeing the whole orchestra, not just one instrument.
Mm.
That's what we're diving into today. This amazing 1995 science paper that, well, it kicked off the ability to monitor thousands of genes simultaneously.
It's hard to overstate the change. Before this paper, I mean, if you wanted to know which genes were switched on in a cell, it was incredibly laborious.
Right.
You had things like northern blots, S1 nuclease assays, maybe RT-PCR later on, but each one only let you look at, a handful of gene transcripts, maybe just one or two.
Like trying to understand a whole city by listening to just a couple of phone calls.
Exactly that.
Yeah.
But then back in the early 90s, this team at Stanford, Pat Brown and Ron Davis were leading it. They asked this really fundamental question.
Which was?
Could we actually quantitatively see the expression levels? Not just presence or absence, but how much? And for hundreds, maybe thousands of genes, all at the same time across different cells.
Wow. What was driving them? Just curiosity.
Well, partly, yes. Basic curiosity about how gene activity is controlled on a massive scale, but also a very practical need. Think about cancer or how embryos develop. These involve complex patterns, right? Lots of genes changing together. They needed a way to see that whole picture.
Okay, so let's get into how they actually did it, this revolutionary idea. The core, as the paper lays out, was this thing called a cDNA microarray. And figure 1, it really shows the first big innovation, robot spotting.
Yeah, the spotting. It's actually kind of amazing when you think about the precision back then. So imagine a standard glass microscope slide.
Okay.
They treated it with something called polylysine. Basically, it just helps DNA stick to the glass. Simple chemistry trick.
Got it.
Then, using a robot arm, they actually built this thing, a high-speed robotic arm. They would precisely place thousands of different DNA solutions onto the slide.
Thousands on one slide.
Yeah, thousands. Tiny, tiny little spots, like microliter scale drops, arranged in this super dense grid, maybe 20 by 20 little blocks of spots. Each slide could hold up to 1,000 unique DNA probes.
And each probe is for a different gene.
Exactly. Each spot represents one specific gene you want to look at. And the idea of printing DNA like this at that density, that was really a first for molecular biology, borrowing from engineering, really.
So you've got this slide packed with tiny dots of DNA for all these different genes. But how do you actually use it to measure gene activity? That seems like the next big hurdle.
Right. And that's the second really clever part, also shown in Figure 1. It's this technique called dual color fluorescence hybridization.
Dual color. Okay, so 2 colors involved.
Precisely. What they do is take 2 different biological samples. Let's say normal cells versus cancer cells, or cells before and after you treat them with a drug.
Okay, comparison.
Exactly. You extract the messenger RNA, the mRNA, from both samples. That tells you which genes are active. Then you convert that mRNA into complementary DNA, cDNA. And here's the key. You label the cDNA from sample 1 with one fluorescent dye, say one that glows green, like CSI3, and you label the cDNA from sample 2 with a different fluorescent dye, one that glows red, like CSI5.
So green tags for sample 1, red tags for sample 2.
You got it. Then you mix these 2 labeled cDNA populations together, and you wash them over the microarray slide.
And they stick to their matching spots.
That's the hybridization part. The labeled cDNA from your samples will bind or hybridize only to the spots on the slide containing the matching gene sequence. And the idea is simple. The more active a gene was in your original sample, the more labeled cDNA you'll have for it, and the more will stick to that specific spot on the array.
Ah, so the brightness tells you something, but why the 2 colors?
That's the genius of it. Because you have both colors hybridizing to the same slide at the same time, you get a direct comparison right there on each spot.
Okay, how does that work?
Well, after you let them hybridize, you wash off anything that didn't stick, and then you scan the slide with a special laser scanner. It measures the intensity of the green fluorescence and the red fluorescence at every single spot.
I see.
The ratio of those 2 intensities, how much red versus how much green light, is coming from one spot that directly tells you the relative activity level of that gene in your 2 original samples.
So if a spot is bright red,
that gene was much more active in sample 2, the red labeled one.
bright green,
More active in sample 1.
and if it's kind of yellowish,
Then the gene was probably active at similar levels in both samples because red and green light together make yellow.
wow, okay. And you're doing this for thousands of spots at once. The amount of data.
It's staggering. From one single slide, one experiment, you could suddenly get, like, potentially 100,000 quantitative measurements. It was just
Yeah.
a flood of information compared to before.
An absolute flood. But just getting floods of fluorescent light data isn't the end goal, right? You need to make sense of it. Which brings us to the software and stats side they mentioned in section 2.3.
Absolutely critical. It's easy to focus on the wet lab stuff, the arrays themselves, but they didn't just invent the physical technology. They also had to develop the tools to actually read and, importantly, interpret the data coming off it.
Like what kind of tools?
Well, first, the scanners themselves to accurately detect those fluorescent signals, but then, crucially, the computer scripts. Custom software they wrote to process all those intensity readings.
What did the software do?
Essentially, it turned those raw fluorescence measurements for each spot for both colors into a huge organized table. A matrix, basically. Numbers representing the activity level or the ratio of activity for every single gene on the array.
So turning the colorful picture into a spreadsheet you can actually analyze.
Exactly. That matrix became the starting point for all the downstream analysis, figuring out which genes changed the most, finding patterns, grouping genes that behaved similarly. It laid the groundwork for, well, pretty much all modern gene expression analysis pipelines.
Okay, so they've got the array, the labeling, the scanning, the software. It sounds amazing in theory, but did it actually work? How did they, prove it? That's section 2.4, the validation part, right? Maybe figure 2 shows some of this.
Yeah, validation was absolutely key. You can't just throw a new technology out there. They needed to show it was sensitive, reliable, reproducible. So they used some well-understood biological systems first.
Smart. Like what?
They looked at plants, actually, how gene expression changes when you expose a plant to light. Some genes turn on, others turn off. They also compared different tissues in Arabidopsis, which is like the lab rat of the plant world. They compared genes active in roots versus leaves.
And figure 2 presumably shows these patterns.
Right. If you look at figure 2 in the paper, you can visually see the differences. For instance, genes everyone knew were involved in photosynthesis. They lit up way more in the leaf samples compared to the root samples on the microarray.
As you'd expect.
Exactly. It showed the system could pick up known biological differences. They demonstrated it could reliably detect changes in gene expression over a pretty wide range, like 50-fold, maybe even 100-fold differences. And importantly, if they repeated the experiment, they got consistent results, so it was sensitive and reproducible.
It really sounds like they nailed it. But what strikes me, looking back at section 2, is that this wasn't like discovering a totally new force of nature. It was more about cleverly putting existing pieces together, wasn't it?
That's a great way to put it. It was an incredible feat of integration. They skillfully combined techniques that were already mature, or at least known in different fields.
Like what?
Well, PCR for making the DNA probes, the fluorescent dyes were available, the surface chemistry for getting DNA to stick to glass, people were working on that, and, really crucially, automation engineering.
The robotics.
Yeah, the idea of using robots to handle tiny liquid volumes, precisely that robotic spotting was already being used in big gene sequencing centers. But Brown and his team saw how they could adapt that specific capability for a totally different purpose. Analyzing gene expression, not sequence, and doing it on glass slides was fundamentally different and much more powerful than the older methods using membranes.
So this 1995 paper wasn't just here's a neat new lab trick. It fundamentally changed the whole approach, the whole philosophy of studying gene expression. Our notes talk about this huge paradigm shift.
Oh, absolutely. It sparked a true revolution. Before this, you were studying maybe one gene, getting a tiny message. This microarray text suddenly let you see the entire transcriptome, all the messages at once. The panoramic news, as the notes say.
From a single sentence to the whole newspaper.
Pretty much. It moved biology from analyzing genes in isolation to looking at the whole system, the whole network of activity. This was really the birth of high-throughput transcriptomics.
And you can see the ripples immediately. This wasn't just a one-off invention. It directly led to other things, right?
Definitely. This foundational work paved the way directly for the next generation of chips. Things like oligonucleotide chips, where instead of spotting DNA, you actually synthesize short DNA sequences directly onto the chip surface.
More precise, maybe.
Yeah, and easier to scale up. And then whole genome chips, covering basically every known gene in an organism. Commercial companies like Affymetrix jumped on this wave, building platforms based on these concepts.
And it wasn't just the hardware, the chips themselves.
No. It spurred huge innovation in the supporting tech, too. Better array scanners, much more sophisticated statistical methods and algorithms for dealing with the data, especially how to normalize those two-color channels properly, how to handle the sheer volume, and things like hierarchical clustering, ways to visually group genes that act alike, that came out of needing to make sense of microarray data.
So it even influenced the math and software side profoundly.
Hugely. Even now, with RNA-seq being dominant, a lot of the statistical thinking, how we compare multiple samples, look at thousands of genes simultaneously, really has its conceptual roots in the problems first tackled with microarrays.
It's wild to think how quickly it spread. You said within 5 years.
Yeah, the adoption was incredibly fast. By the early 2000s, labs everywhere were using microarrays for all sorts of things.
Like what? Give us some examples.
Okay. Classifying different types of cancer. Tumors that look similar under the microscope might have very different gene activity patterns, and microarrays could reveal that, potentially guiding treatment.
Huge clinical potential there.
Right. Also, drug development, testing how a potential drug affects gene expression across the whole genome to understand its effects and maybe predict side effects. Toxicology, studying stem cells, how they differentiate into muscle or nerve cells, watching those gene programs change over time. Even ecology, looking at how wild organisms respond to pollution or climate change at the genetic level.
Wow, really diverse applications.
Incredibly diverse. The paper itself, by 2002, had something like over 5,000 citations. That just shows you how quickly and broadly the scientific community grabbed onto this.
Of course, you mentioned RNA-seq becoming dominant later, so microarrays weren't perfect. They had limitations.
Oh, for sure. No technology is perfect, especially a first-generation one. Over time, people realized certain limitations. Things like hybridization specificity. Sometimes a labeled cDNA might stick weakly to a spot it wasn't perfectly matched to, creating noise.
Crosstalk?
Kind of, yeah. Also, the dynamic range detecting both very weakly expressed genes and very highly expressed genes accurately on the same array could be challenging. And comparing data strictly between different experiments, especially on different platforms or batches of arrays, that could be tricky due to technical variations, these batch effects.
Right.
RNA-seq, which came later and directly sequences the RNA fragments, it managed to overcome some of those specific issues, offering better dynamic range and less background noise in many cases.
But the core idea.
But the core concept from that 1995 paper, the idea of reading the whole transcriptome at once, getting that global view that absolutely stuck. That's the legacy. RNA-seq is just a different, often better way of achieving that same fundamental goal pioneered by microarrays.
Something else that seems really striking about this story is how it happened. The notes mention this university garage-style collaboration and an openness.
Yes. That's another fascinating part of the story. The environment at Stanford's Beckman Center was apparently quite unique. It brought Ron Davis's lab, with all this expertise in automation and engineering from the genome sequencing world, right next door to Pat Brown's lab, focused on gene expression.
Serendipity.
Maybe a bit of design serendipity. And you had the whole Silicon Valley startup culture nearby, encouraging people to build their own tools, take risks. It was a small team, just 4 key people listed on that first big paper. Mark Schena and Dari Shalon, who did a lot of the crucial bench work and optimization, plus Brown and Davis leading the charge.
And the sharing part. That seems unusual, especially for potentially valuable tech.
It was remarkable for the time. Pat Brown, very early on, made the decision to basically give the technology away. They put the design files for the robot spotter, the software they wrote, even the lists of gene sequences they used as probes, all up on a website, freely available.
Really? Why?
The idea was to get it out there, let other labs build it, use it, improve it. They wanted to accelerate the science, and it worked. Labs all over the world started building their own microarray systems based on those shared plans. It created this informal expression chip alliance, as the notes call it. That open approach was absolutely fundamental to how quickly the field took off.
That's a really different model from keeping things proprietary. It highlights the people involved, too.
Definitely. You had Pat Brown bringing this cross-disciplinary perspective. He had physics and medical training thinking about printing genes. Ron Davis, the engineer, the automation guru who knew how to physically build the machines needed. And then Mark Schena and Dari Shalon, the experimentalists who really dialed in the tricky bits, especially the surface chemistry, getting the DNA to stick properly and be available for hybridization. That was not trivial.
A perfect storm of different skills.
Exactly. It really underscores how breakthroughs often happen at the intersection of disciplines. You needed the biological question, the engineering know-how, the chemistry, the computation, and that culture of collaboration and sharing to make it fly.
So thinking about the bigger picture.
Yeah.
How did this 1995 paper really inspire other research, beyond just making microarrays?
Well, in several key ways, I think. First, it was a huge advertisement for the power of interdisciplinary science. It showed people that biologists, talking to engineers, talking to computer scientists, could solve problems that none could solve alone.
Break down the silos.
Right. Second, it highlighted the value of adapting technologies, taking that robotic spotting from sequencing and completely repurposing it. It encouraged people to look at tools in other fields and ask, hey, could we use that for our biological problem?
Resourcefulness.
Definitely. Third, that open sharing model, while not always replicated, it really did foster a sense of community and rapid collective progress in early genomics. It set a precedent, even if an idealistic one. Fourth, the sheer success of microarrays in generating massive data sets fundamentally shifted how biologists thought. It pushed them towards genome-wide thinking, systems-level thinking, rather than just focusing on one gene or pathway.
Paving the way for other omics.
Absolutely. It created the appetite and the analytical frameworks for proteomics, metabolomics, all these other fields trying to measure large sets of molecules. And finally, those diverse early applications, cancer, plants, drugs, they showed the immense breadth of questions you could tackle with a global view. It inspired researchers in countless areas to think about how understanding the entire pattern of gene activity could unlock secrets about complex diseases or biological processes.
It really is incredible to trace the lineage from that one paper, how it just opened up so many doors.
It truly is. And it makes you think. This ability we now have, largely thanks to that initial breakthrough to simultaneously watch thousands, now millions of molecular events, it's changed biology forever.
Right.
But it also makes you wonder, what's next? What are the next generation technologies going to let us see? What even deeper insights into the complexity of life are waiting just around the corner if we can just figure out how to measure them all at once?